Here is our replication review video. So essential understandings from IB, genetic information in DNA can be accurately copied, which is super important so that we can send information on to daughter cells. It's also semi-conservative. So we're going to look a little bit at what that conservative piece means right here. So there were two scientists, Mendelssohn and Stahl, who did an investigation with DNA replication. They started DNA nucleotides that had nitrogen-15. This is a radioactive heavy version of nitrogen-15. So all of the original strands started with nitrogen-15, 15, 15, 15. Everybody started with 15. Then, when replication began to happen, only nitrogen-14 was available. So all the nucleotides that were being used to make the new DNA was nitrogen-14, a little bit lighter. Nitrogen always has 7 protons, 15 has 8 neutrons, 14 only has 7 neutrons. This guy is heavier. So if replication was conservative, which means that the whole thing got saved, then we would have one strand that was 15, 15, and the new strand would be that new nitrogen 14, 14. We would expect to see in the centrifuge. So we took the DNA and we spun it in the centrifuge tubes. So the 15 should be heavier. It's going to go down here. 14 is lighter. It's not going to sink as far in the tube. So 14, 15. These guys are kind of turned sideways. So this is 15. This is 14. Had replication been found to be conservative, then I should have gotten, after the first generation of replication, a 15, 15 and a separate 14, 14. So if we look at generation number 1 here, we should have had a 15, 15 and a 14, 14. We have neither of those. We have half and half. So semi-conservative is a better prediction for we got half and half, because each one is 14, 15, 15, 14, half and half. But dispersive, if everything just got like broken up and put back together, also could have led to a 14, 15, 14, 15, everything in the middle. So we look at that second replication. Semi-conservative plan would give us 2 pieces that were 14, 15 and 2 pieces that were 14, 14. Whereas here we'd have, again, everything mashed up in the middle. So if we look at, this is almost generation 2, there are 2 separate bands, one halfway between 14 and 15 and one at 14. So this is our 14, 14 band. This is our 14, 15 band. This guy are these 2 pieces that are all nitrogen 14. And then this band was our 2 pieces that were 14, 15, 14, 15. So this investigation allowed Mendelssohn and Stahl to determine that DNA replication is semi-conservative. Each new piece of DNA is half old DNA. Replication occurs under the control of many, many, many enzymes, more than what's in the list here, but these are the ones that we need to know. These are the ones that we're going to talk about. So, helicase is an enzyme that's going to unwind the double helix and break the hydrogen bonds between complementary base pairs. So here's our helicase unwinding the helix and breaking the hydrogen bonds between the complementary base pairs A's and T's, C's and G's. Topoisomerase, which is also called gyrase, is here. It's going to relax the supercoiling of the DNA, allow it to not get knotted up as it untwists. So topoisomerase or gyrase and helicase are helping to unwind, break the knots, unzip the DNA. The next enzyme that comes in is DNA primase. So primase is actually going to put down, this is weird, a small strand of RNA, and this is on both strands. This is not just the lagging strand, on both pieces of DNA. Both of those strands that have just opened up, we're going to start with some DNA primase. That's going to put down a small strand, 10 to 15 A's, U's, C's, and G's, of RNA because DNA is a little high maintenance and refuses to start. We cannot start a strand with DNA. We can start with RNA, and then we can put DNA on the RNA. We just can't start with DNA. So DNA primase is going to put down a short strand of RNA to get it started on both the lagging and leading strands. Both of them need a little bit of RNA to get started. DNA polymerase is technically 3, but we don't have to know that. One of the DNA polymerases is going to attach to the primer and then attach DNA nucleotides. So now A's, T's, C's, and G's attach the DNA nucleotides to the primer in a 5' to 3' direction. But in biochemistry, we have to count the carbons. So I take the 5' carbon of the new guy and I stick it on to the 3' carbon of the guy that's already there. The new nucleotide that we're adding, we take the 5' carbon, 5 next to the phosphate group, and we stick it onto the 3' carbon of the sugar molecule of the previous nucleotide. Everything goes in a 5' to 3' direction, every single time. This happens in both the leading strand and the lagging strand. Here's just a close-up view of that idea. So this is the 3' carbon of the nucleotide that's already there. This is the 5' carbon of the new guy. We're going to attach that to that just like that. We always go in a 5' to 3' direction. Always, always, always. So because we can only go in a 5' to 3' direction, we end up with leading strands and lagging strands. The leading strand, we can make this new DNA continuously. As the DNA opens up, it can just keep going, keep going, keep going in that direction. Our lagging strand cannot go that way. It has to go that way. And so what happens is we open up the DNA and we make a strand like that. And then we open it up a little more and we make a strand like that. We open it a little bit more, and then a little more, and a little more, and a little more. And we just keep going piece by piece backwards. Each of these pieces is called an Okazaki fragment. Okazaki fragments are found on the lagging strand because polymerase can only attach new nucleotides in a 5' to 3' direction. Those primers, the RNA primers, need to leave before we finalize our new DNA. And so we have another enzyme called exonuclease. It's going to come in and remove the RNA primers. Another polymerase, another DNA polymerase, is going to come in and replace the holes where the RNA used to be with new DNA. And then a ligase, another enzyme, is going to come in and glue together all the fragments. So ligase is going to glue here and here and here. Everywhere we have a little chunk, ligase is going to glue together. This is far more common between those Okazaki fragments, but it can also happen on the leading strand because sometimes you can imagine DNA is like this long. It would be silly of us to start here and go entirely down the whole strand with just one enzyme. So what usually happens is even on the leading strand, we'll have multiple starting places and we'll copy the DNA, copy the DNA, copy the DNA. And now these fragments, even on the leading strand, so not Okazaki fragments, just pieces of DNA, can also get bound together by ligase. We can take advantage of this in the lab with a piece of biotechnology called PCR. PCR stands for polymerase chain reaction. So chain reactions is when we just keep repeating things over and over and over. We use the products of one thing to make the next thing happen. We use the products of that for the next thing to happen. We're going to use polymerase, that enzyme that builds new DNA, to make it happen. There are 3 stages: denaturation, annealing, and elongation. So in denaturation, we heat things up. That high temperature is going to break the hydrogen bonds between the complementary base pairs of the DNA. And then we anneal with a primer. So we have some specific piece of DNA that we made in the lab. We're going to add it to our denatured DNA. So we're going to add a primer we're going to glue it or anneal it onto our template strands. And then we go through extension, and during extension, we're going to use that polymerase enzyme to build new DNA copies of the chunks of DNA that we started with our primer. So this is one particular cycle. Each cycle takes about 2 minutes. I can repeat and repeat and repeat, which means that just about every 2 minutes, I'm doubling the amount of DNA in the sample. So here's just showing I've got my template DNA. This is the gene that I'm particularly interested in. I'm going to denature it so that it gets split in half. I'm going to add a primer, and then we're going to extend, and we're going to copy those two pieces that originally here and here. And then I heat it up again, and those two pieces get separated. We copy, they get separated, copy, and then we do it again and again. And so every 2 minutes, I've doubled the amount of DNA in my sample, which gives me lots of DNA to run tests. We can do this at these crazy high temperatures because we have stolen an enzyme from a bacteria called Thermus aquaticus, which is literally hot water. So TAC, this TAC polymerase, allows us to carry out DNA replication at very high temperatures, so we can make it happen really fast in the lab. That's the end of DNA replication review. Let me know if you have questions.